Hi, I'm Jordan, and my proposal in the beginning of my thesis is assessing how extracellular vesicles from human milk influence a Keiko-2 cell model of the human intestine. Briefly, the contents of this presentation are as follows. The first half being background and rationale for this proposal, followed by the central hypothesis, the experimental design, methods, expected outcomes, and lastly, any future directions for this project. To begin, I'm going to start off by talking about human milk, which is widely regarded as the gold standard for infant nutrition for the first 6 months of life. The American Academy of Pediatrics, World Health Organization, and United Nations International Children's Emergency Fund, or UNICEF, all recommend human milk to be the sole food for infants up until the first 6 months of life. This is because human milk provides all the essential nutrients and beneficial bioactives for infant growth and development. Bioactives are primarily non-nutritive compounds that elicit positive effects on health. Some bioactives in human milk are included on this slide, such as vitamins and minerals, human milk oligosaccharides, probiotics, microRNA, and extracellular vesicles. It is thought that these bioactives are responsible for the improvements in health seen in infants fed human milk over those who are formula fed. It's important to determine the role of these components in human milk because a large portion of infants are not breastfed. Due to challenges with technique, pain, social pressure, insufficiency, inability to obtain human milk from donor banks, or it's simply just choice, a large portion of infants are not fed human milk. As you can see from data on the left collected by the CDC in 2017, only about 80% of infants are ever breastfed. Additionally, out of those infants who are breastfed, less than half of the infants are breastfed for even 3 months, and only about 30% reach the duration recommended by the World Health Organization's previously stated, and that is shown on the right. Infant formula contains a profile of macronutrients similar to human milk, with some small differences in protein levels and source of fats. But since we are still discovering many of the bioactives' role in human milk, a lot of them are not implemented in infant formula. Most commonly, the bioactives added to infant formula are probiotics and oligosaccharides. However, this has not been enough to bridge the gap between infant formula and human milk. And bioactives, such as the one listed on the slides, like immunoglobulins, HMOs, white blood cells, and peptides, are still only currently showing their effects in human milk. This has led to some health disparities in infants who consume infant formula and those who consume human milk. Infants who are fed human milk see more controlled weight gain throughout the first few years of life, reduced occurrence and severity of gastrointestinal infections, higher Bayley scores of infant cognitive development, and in preterm infants, a significantly lower risk of necrotizing enterocolitis, or NEC. NEC is a deadly condition in which bacterial disease destroys the intestine. Human milk has such a protective effect against NEC that if an infant is born premature and consumes infant formula, it is a risk factor for developing NEC. Unfortunately, in full-term infants, intestinal health is still a concern. After delivery, infants have an immature, permeable, uncolonized intestine and a comparatively weak immune system to adults. On the left, you can see an example of increased permeability between enterocytes or intestinal cells, which allows for bacteria to permeate the intestine and reach the bloodstream, which can cause illness. This particular illustration is for leaky gut and therefore is an exaggerated version of what is seen in infants, but serves as an illustration for the tight junction gaps. On the right, on image B, you can see the immaturity of enterocytes, which are the purple ovals. These do not possess fully developed crypts or villi and would not be fully functional. On the image above, the enterocytes have finger-like projections, which are the villi, and the crypts, which are the small valleys. GI diseases in infants can impact growth, development, and even be deadly, so it is critical everything is done to prevent these in infants. One bioactive of interest that may be responsible for some of the protective effects seen in human milk are extracellular vesicles, or EVs. EVs are nanoparticles, typically between 50 to 10,000 nanometers, that carry cargo such as proteins, DNA, mRNA, microRNA, and lipids. EVs are differentiated from other particles by the presence of what the Journal of Extracellular Vesicles call EV-enriched proteins. These include transmembrane proteins, cytosolic proteins, and EV co-isolated structures. The presence of each of these protein types is required for the validation of EVs in any product. There are three different classifications of EVs, which are exosomes, microvesicles, and apoptic bodies. These are broken up based on biogenesis and size. Microvesicles range from 50 to 10,000 nanometers in diameter. Exosomes range from 30 to 150 nanometers in diameter, and apoptic bodies range from 50 to 5,000 nanometers in diameter. Microvesicles are formed from the outward budding of the plasma membrane, while exosomes are formed from the inward budding of early endosomes. And apoptic bodies are formed when the membrane of a dying cell blebs and the vesicles break off, containing its cellular components. All of these EVs are capable of bringing cargo to other cells as a means of communication. EVs are encompassed by a lipid membrane, which both protects cargo and makes it easy for them to fuse with the recipient cells. Once EVs are taken up by the recipient cell via a variety of methods, they release their cargo, which is able to then elicit the desired effects on other cells. EVs are produced from all eukaryotic cells. However, most of the available literature on EVs focus on endogenous EVs produced from either stem cells, blood, urine, or cerebrospinal fluid. These endogenous EVs have shown positive effects on disease and health, with stem cell-derived EVs improving outcomes for chronic kidney disease, increasing sensitivity to cancer treatments, and promoting angiogenesis. Because of these promising effects, the endogenous EVs have been showing clinical trials on patients with immune disorders, neurological disorders, cardiovascular disease, cancer, kidney disease, and even COVID are underway. Additionally, there is some preliminary research suggesting that EVs isolated from patients can be used as biomarkers in diseases like cancer, Alzheimer's, and dementia. While there's becoming more literature on endogenous EVs, dietary EVs we consume still remain fairly unexplored. So far, though, EVs have been discovered in foods such as honey, ginger, berries, carrots, bovine, and human milk. Studies determining the effect of these EVs have found data supporting bovine and grape EVs as having positive effects on mice health, and honey, grapefruit, ginger, and carrot EVs as being able to modulate cytokine responses of macrophages and stem cells, suggesting anti-inflammatory effects. These results from dietary EVs suggest that there may also be beneficial effects from human milk EVs. Luckily, some researchers have begun to look into this, as human milk EVs have been shown to survive digestion and are able to be taken up by CACO2 cells, an intestinal model cell. Additionally, human milk EV-treated gingival cell and rodent IEC cells have shown faster healing to damage from hydrogen peroxide in physical cuts. Lastly, in rodents who underwent an experimental method for developing NEC, pictured on the image at the top right, rodents who were fed human milk EVs had a significant reduction in NEC compared to the control. These studies show the beginning of the potential human milk EVs have on health. With the available information on human milk, infant health, and the current state of EV research, we hypothesize that human milk EVs will improve proliferation, maturity, and permeability of intestinal cells, and that this may translate to better intestinal health of humans in later studies. We aim to test this hypothesis through a series of experiments that will assess viable cell counts, morphology, maturity, and membrane electrical resistance. This will be done using the equipment available in Keck Center on Chapman's campus. To form our intestinal model, we'll be using CACO2 cells number HTB37 from the American Type Culture Collection, or ATCC. These cells are derived from human adenocarcinoma cells and have been modified to express the characteristics of intestinal cells. This strain of cells is commonly used in scientific research and has proved to be a reliable model of the human intestine. According to ATCC recommendations, these cells will be grown in a medium comprised of 80% Eagle's minimum essential medium and 20% fetal bovine serum, which are pictured on the right of this slide. Initial seeding of these cells will be done in 25 centimeter squared culture flasks at a cell density of 4 × 10⁵ cells per centimeter. These cells will be allowed to grow until they reach 80% confluence of the growth plate, at which point there will be passage, which removes 90% of the cells and places the remaining 10% of the cells in a new culture flask for growth. This will be repeated until passage 20, where the cells will be allowed to reach full confluence for 3 weeks to allow for the formation of the monolayer. The monolayer can also be formed by the use of cell inserts. However, our lab has an established protocol for cell differentiation by allowing them to reach confluence, and literature suggests no difference between the two methods. Before the use of these cells in our experiments, they will need to be examined for the presence of a monolayer to ensure cell maturity, which is pictured on the right of this slide. And this will be done using microscopy on the full culture plates of the cells. The human milk samples we obtained from a company called Fonterra that's located in New Zealand. Our human milk samples will be aliquoted in 2 milliliter microtubes and centrifuged at 2000 G for 10 minutes. This will form a fat globule layer at the very top, which will be discarded via a metal spatula. Next, the samples will be centrifuged at 12,000 G for 30 minutes. This will allow the cell debris to form towards the top, and this will again be removed with a metal spatula. The remaining supernatant will then be filtered with a 0.45 micrometer cellulose syringe filter. After this, the supernatant will be combined with a reagent called ExoQuick TC in a 5:1 ratio. This will then be incubated overnight in a fridge at 4 degrees Celsius. This will allow for the ExoQuick and the supernatant to begin to separate. Then this mixture will be centrifuged at 5,000 Gs for 30 minutes, and an EV pellet will form at the bottom of the microtube. After the supernatant is discarded, the EV pellet will be resuspended in phosphate buffered saline and will be ready for use in our experiment. After the filtration step on the previous slide, there is another way in which it is possible to separate EVs from the rest of the components in our milk sample. Using a method called ultracentrifugation, a process in which vials of our sample are spun at 100,000 Gs, EVs are able to be separated based on their density. However, it takes a larger amount of sample, with some methods requiring up to 160 milliliters of samples to do this, and precipitation-based methods such as ExoQuick's TC has been shown to isolate a larger amount of EVs from a similar sample. After the EVs are separated and our KCO2 cells are fully matured, the incubation of the cells with their respective medium can begin. There will be 3 groups: an experimental, positive control, and negative control. The experimental group of cells will receive media comprised of 30 micrograms per milliliters of human milk EVs and complete growth medium. Our positive control will have medium that contains 2% dextrans sodium sulfate, which is a stimulus of inflammation, along with the complete growth medium. And our negative control will only have complete growth medium. All of these groups will be incubated with their respective media for 24 hours, and experiments will be carried out with 3 replicates in each group. After all groups have been incubated for their respective time point, testing can begin to determine the effects human milk EVs have on these intestinal cells. First, the monolayer's electrical resistance will be determined by the use of transepithelial electrical resistance, or TEER. This measures the difference in resistance between the culture media and the cells to give the resistance value for the membrane of the cells only. This is done by placing an electrode in a 2-part plate, with one part containing only our culture media and another part containing our cells with the culture media, as shown in the middle image. Value obtained from this can be used as an indication of the strength and permeability of the cells in the monolayer. We have chosen this method over examining the levels of a specific tight junction protein, such as agglutinin, because it will give a more broad pictures of how the cells in the membrane are doing. And being too specific at the start, we may miss alterations and levels of other tight junction proteins. After confirming that there is indeed a difference in TEER measurements in our experimental groups, it would be a good idea to go back and examine which protein levels were affected. Next, our KCO2 cells will be observed for maturity and their morphology. This will be done via digital microscopy. To observe the cells, they will first be fixed. This will be done by a series of dehydration treatments where the cells will undergo a 50, 75, 96, and 100% ethyl alcohol bath. After this, the cells will be cleared of the ethyl alcohol using 3 baths of xylene, which are 1 minute each. After that, the staining is ready to begin, where we will use hematoxylin and eosin. After that, we will embed the cells in paraffin blocks and section them into 5 micrometer slides. After this, they will be ready to be observed with our digital microscope. We'll be examining them for the villus height and crypt depth. This will give us an idea of the maturity of the cells. In this slide, we can see an example of the morphology we expect to see on the left and the staining color we expect to see from the hematoxylin and eosin staining on the right. Lastly, the viable cell counts for all of our groups will be measured. This will be done using trypan blue and a specialized cell counter called the Countess Automated Cell Counter. A 1 milliliter sample of our cells suspended in growth medium will be homogenized, and then a 50 microliter drop of our cells and a 50 microliter drop of trypan blue will be added to this slide chamber. The trypan blue only enters cells which are negatively charged, and so it differentiates between alive and dead cells, which we can see in the picture in the middle. This allows for the Countess machine to quickly tell us how many viable cells we have in our drop, which we are then able to use and extrapolate out to know our total amount of viable cells in our known volume. Viable cell counts will be measured in this manner for 21 days. Each day, the number of viable cells and total number of cells obtained from the Countess machine will be plotted on a graph to show the growth of the cells after treatment, as well as which growth phase they are in. Our data will be analyzed using RStudio, and we will do 2-tailed ANOVA analysis and a mean comparison test called the Kruskal-Wallis test on our results. The significance for all of these statistical tests will be p is equal to less than 0.05. For our experiments, our expected outcomes are as follows. First, increase monolayer electrical resistance in the experimental group compared to both control groups. This would indicate that the permeability of our cells has decreased and that the tight junctions are intact. Secondly, increase villus height and crypt depth in our experimental group in comparison to both of our control groups. This would indicate that human milk EVs promote the maturity of intestinal cells. And lastly, an increased number of viable cells in our experimental group compared to the controls. This would indicate that human milk EVs promote cell proliferation. All of these results will provide support that human milk EVs are beneficial for the growth and development of intestinal cells. While this data would only be for an in vitro model of our CACO2 cells, further research on human milk EVs for either use as supplements for infants or to be fortified in infant formula could greatly benefit infants. Further research for these experiments would include examining more mechanisms where human milk EVs impact intestinal cells. This could include examining protein levels of the cells or how molecules are trafficked. Human milk EVs could also be used in further studies with rodents to give better insight into how a full body system would react to the implementation of human milk EVs. And lastly, if no negative effects were seen in animal models, human milk EVs may be used in clinical trials to determine how they affect human intestinal health. This would provide a great benefit to infants, especially those who are not fed human milk. This may be used to prevent illness and disease, which is a priority for agencies such as the USDA. Let me know if you have any questions. Thank you for listening, and here are my references.